Hi everyone, this is lecture 6. This is going to be an extension of DNA replication, but focused on the techniques of PCR and DNA sequencing, which are both extensions of DNA replication. Alright, the material from this is kind of scattered throughout the textbook. It's found in chapters 9, 10, and chapter 8. There are small segments. There's a nice figure in chapter 9. In addition, there's some information on the learning module, and there's plenty of information online about PCR. We have no class on Monday because of Labor Day, so there will be no quiz for Monday. The next quiz will be due a week from now on Friday. I'll give you more information when we get closer to that. So happy Labor Day. All right, let's get started. First off, a couple of quiz questions. Number 1, DNA helicase is used in PCR to separate the two DNA strands from one another. True or false? Number 2, which of the following is or are needed for the PCR technique to work? There may be more than one correct answer. Hint, hint. Number 3, dideoxyribonucleotides are useful in DNA sequencing because and 4, given the following region of genomic DNA, which pair of primers would amplify the majority of this sequence? Now, these are all questions that will help guide your discussion and learning in today's lecture, and these are questions that will show up on the exam, on the quiz next Friday, and potentially on the exam. So today we're going to be talking about PCR, which is simply an application of DNA replication. PCR stands for polymerase chain reaction. A chain reaction is simply a reaction where the products of one are used as substrates for the next. And so in that sense it keeps going on its own once you start the initial steps. This technique was invented by Kary Mullis in 1983, who went on to win the Nobel Prize for this discovery. Pretty awesome. PCR can amplify DNA over 1 million-fold. And this is pretty quick. It happens in about an hour, but of course on TV it has to happen within a shorter amount of time. So if you're watching crime shows and so on, you'll see PCR sometimes being done. This technique is done a lot in forensics and other applications because it can take a small amount of DNA and amplify it to a large amount so that you can analyze it by standard molecular and genetic biology techniques. Let's go through the steps of PCR. The first one is the 2 strands of the double helix have to be denatured. This means the hydrogen bonds have to be separated. As opposed to replication in vivo, we can just simply heat the DNA with high temperature, in this case 95 degrees Celsius, and 15 seconds is plenty of time. At that point then, small oligonucleotides, which are the primers, will hybridize via hydrogen bonding to the DNA template. These primers are not unlike the ones that would be used in vivo, except they are synthesized chemically in a lab, and so they can be designed for specific genes, as opposed to the primers that are used in replication in vivo, which are simply randomly generated DNA and oligonucleotides. Another difference is that these primers are DNA primers because they are more stable and they're easier to synthesize. The primer annealing or hybridization happens at about 55 degrees. We need 2 primers. One's in turquoise here, that's called the forward primer, and one is in yellow, that's the reverse primer. These 2 primers are shown with arrows to indicate their directionality. You'll see here at the bottom. The arrow points in the 5 to 3' direction. Notice that they're hybridizing to the template in the anti-parallel direction, so the template is in white, and it's going from right to left, 5' to 3'. These 2 primers, the forward primer and reverse primer, are essentially pointing towards each other. That is the region between those 2 primers that will be amplified. Remember, your genomic DNA is very large. PCR only amplifies a very small portion of it. The next step is we need a special enzyme. This is a special DNA polymerase that was isolated from a thermophilic bacteria. That's a bacteria that lives in hot springs. That bacteria is called Thermus aquaticus, and so we abbreviate the bacteria's name with TAC, T for thermus, AQ for aquaticus. And the DNA polymerase from that enzyme is heat stable, so it can withstand even boiling temperatures, which is pretty amazing. TAC polymerase will bind to the primer and template and will extend given the right temperature, which is about 72 degrees. It works at about 1 minute per kilobase of DNA and makes new DNA shown here in purple. These products then have now been generated will be denatured again, as shown here. So you've gone from 1 template to now 2 templates. We've just doubled the amount of DNA that we have. In order to double again, we need to repeat the steps. So the DNA is denatured. Primers are bound. So we repeat the 55 degrees. The forward and reverse primers now are binding. And the TAC DNA polymerase is now extending. So only some of the templates are shown here for clarity. Notice, however, that some of the templates have ended here at the forward and the reverse primer. So the subsequent products will only be generated that extend only to the forward and reverse primer ends. Now we have gone from 1 to 2 DNA molecules. Now we're going to go from 2 to 4, by repeating the denature, annealing, extension. And from 4, we go from 2 to 4, and then from 4, we go to 8. And so on. Every cycle that we repeat is a doubling of the amount of the DNA that we get. And so you can do the math, but after 20 cycles or so, the DNA will be amplified 1,000,000 times. The power of exponential amplification. Now, another extension of DNA replication is so-called DNA sequencing. This is how we can read the DNA of our genomes. Now, the way this works is it also uses DNA polymerase. However, it uses a special modified base, which I'm going to talk about. This technique was originally invented by Fred Sanger, who was working in the UK, and this is called chain termination sequencing. And for this, he won the 1980 Nobel Prize in Chemistry, and this was his actual second Nobel Prize. He also did work on being able to read the sequence of proteins. The special nucleotide that's required for DNA sequencing here is called dideoxynucleotides, abbreviated little d NTPS. Dideoxynucleotides. And the structure is shown here. Notice that instead of an OH, a hydroxyl group, on the 3' carbon of the sugar, there is no hydroxyl, there's just simply an H. Hence the dideoxy, di in this case means 2, because 2 of the carbons do not have hydroxyls. This prevents the formation of the phosphodiester bond, as you can imagine. Let's see how this is important. These dideoxynucleotides are fluorescently labeled, and they are shown here in different colors. A is shown in green, C is in purple, G is in yellow, and T is in red. These are fluorescently labeled. Fluorescence is a small molecule that's added to the base, and when you shine light on it, it will fluoresce in a certain color. Add these with a primer that you have chemically synthesized and your DNA template, and these nucleotides will be added in limiting amounts, will be added to the ends of that growing DNA. Now, once they have been added, the polymerization is ended because it's a chain terminating nucleotide, and so there is no additional extension of the DNA. DNTPs are added at low concentrations. They have to be added at low concentrations along with all the other nucleotides. So only occasionally will these bases be added. So for example, one strand, if there is a C in this position, then this is the one that the dideoxynucleotide was added. But you'll notice in this one down here, the second one down, the C that was added was the normal DNTP. So extension could keep going. In this case, though, this one incorporated an A, preventing further extension. However, in this third one down, the C was added as the normal base, the A was the normal base, and only the T was the modified base that entered the chain. Now, in this sense, on average, every base has been represented, and so a machine can come by and read the sequence of these bases. DNA polymerase replicates this template, but stops whenever the DTN-TB is added. When you have these different sized DNA molecules now, they can be separated by electrophoresis. That means you run these DNAs through an electric field and they will separate by length or by size, so that the large DNAs will be retarded in the gel electrophoresis and the small fragments will go through quicker. They separate based on size, and you can then read these with fluorescent tags. Here is an example of sequence. So you have yellow C at the top, the short fragment, all the way to the large fragment, which has got this G in the final position. This is flipped around on the bottom here because we're reading from short fragments to large fragments, bottom to top. And the way that the machine reads this out is with a chromatogram, which is these peaks of different fluorescent labeled colors. There's an awesome animation. We'll take a look at this in class, but also I encourage you to take a look at it. It's from the DNA Learning Center, and it's on cycle sequencing. Just simply Google DNA Learning Center cycle sequencing, and you will find that animation. It'll do a much better job of explaining it than I can in the lecture. DNA sequencing is extremely useful. You can do all kinds of things with it. You can read genomes, you can identify new organisms, you can do molecular phylogenies, that's speciation, you can do genotyping to try to figure out what mutations you may have. This is an example of a study where they were looking at people who were homozygous or heterozygous at a particular position in their DNA. And you'll notice here in this chromatogram, these are represented by different length DNAs with each having a different chain terminating base. Notice that in this position here in the middle, there was an A in this person's genome, and only an A. That means they are homozygous for A. Both of their chromosomes had an A in that position. Whereas this individual down on the bottom was heterozygous, and you can see that because the green and the black showed up. The black in this case is representing the G. So this is one example of how DNA sequencing can be used, but there are, of course, many, many others. Okay, well, that is all for now, so I will see you in class.